To prevent chronic disease, medical science first requires a metric for the physical decay that precedes it. We need a method to quantify biological age before any clinical symptoms can ever manifest. Researchers rely on aging clocks to meet this need. These are computational machine learning models engineered to calculate the exact level of biological wear and tear within an organism. Currently, epigenetic clocks serve as the scientific gold standard in this field. They are heavily utilized because they yield remarkably high predictive accuracy across multiple species. These epigenetic models generate their predictions by tracking highly specific changes in DNA methylation patterns as an organism moves through its lifespan. But this methodology has a critical limitation. Methylation shifts occur largely as a quasi-stochastic drift over time. This makes it exceptionally difficult to interpret the biology behind the math, or to identify which specific cellular functions are actually failing. Transcriptomic clocks offer a theoretical solution to this interpretability problem. Because they measure active gene expression through RNA sequencing, they provide direct, measurable insight into actual tissue function. Historically, however, gene expression is highly sensitive to environmental factors. This introduces extreme variance and noise into the data, preventing transcriptomic models from achieving the predictive accuracy of their epigenetic counterparts. LivAge is a newly engineered Murine transcriptomic clock explicitly designed to solve this environmental noise problem. The model operates by analyzing bulk hepatic or liver RNA sequencing data extracted exclusively from healthy control mice. This schematic maps the three-stage engineering architecture of the tool. The raw data is processed through rigorous curation, algorithmic filtering, and specific mathematical transformations. The strictness of this pipeline results in an R-squared statistical accuracy of over 0.9. At this threshold, the model successfully matches the predictive precision of industry-standard epigenetic clocks. LivAge demonstrates that a strictly controlled data architecture allows transcriptomic models to achieve high predictive precision without sacrificing biological interpretability. The raw material powering this model is a massive transcriptomic atlas. It was built by aggregating 432 healthy liver samples from C57BL6 mice. These samples are strategically distributed across a chronological range from 1 month to 30 months of age, ensuring the data captures a complete murine lifespan. The initial architectural hurdle is sheer volume. While thousands of genes are sequenced in these samples, the vast majority represent biological noise rather than a reliable aging signal. To strip away this noise prior to any machine learning, the researchers implemented a differential expression analysis step to act as a strict biological filter. This volcano plot visualizes that filter in action. By utilizing a false discovery rate threshold, the analysis isolates only the specific genes definitively correlated with chronological aging. This exposes distinct clusters of gene activity, starting with the heavily downregulated pathways shown here in blue. When mapped functionally, these specific downregulated clusters correspond directly to the repression of oxidative phosphorylation and fatty acid metabolism. Conversely, the upregulated clusters in red map directly to an increase in interferon signaling and immune activation. These scatter plots confirm that these abstract transcriptomic signatures correspond to physical cellular shifts. Over time, there is a clear, measurable decline in the overall hepatocyte population. As the hepatocytes decline, they are replaced by a proportional and rapid infiltration of pro-inflammatory Kupffer cells. Aggressively filtering for these specific expression changes anchors the predictive model to verifiable biological realities like inflammaging. This tie between the math and observed tissue decay allows the model to bypass the interpretive limitations of epigenetics. With the biological signal isolated, the data is fed into the core predictive engine chosen for LivAge, elastic net regression. Elastic net is uniquely suited for processing RNA sequencing data because it optimally balances strict feature selection with high-dimensional regularization. However, when this model is applied directly to aggregated multi-study RNA data, the resulting age predictions remain highly erratic. This error stems from two distinct sources: technical batch effects generated by different sequencing labs, combined with the inherently nonlinear trajectory of biological aging. To solve this, the researchers executed a critical architectural pivot. applying dual mathematical transformations to the data before it ever enters the elastic net model. This performance matrix measures the success of these adjustments. The first is a Box-Cox transformation, applied to the age variable to account for nonlinear trajectories. The second is a U-gene normalization, applied to the gene expression matrices, which acts to flatten cross-study technical noise. By tracking the Box-Cox row and the U-gene column to their intersection on the heat maps, the impact becomes clear. This exact combinatorial transformation drives the mean absolute error to its lowest point, while maximizing the R-squared metric to its absolute peak value. This mathematical pre-processing isolates the age-associated transcriptional signal, locking in a predictive 268-gene architecture. For an aging clock to be valid, it must isolate biological wear from the simple passage of time. The foundational metric used to prove this capability is known as age deviation, or age-dev. Mathematically, age-dev is defined as the beta coefficient gap between the model's predicted transcriptomic age and the animal's actual chronological age. To validate the model, LivAge was tested against murine progeroid models, mice bearing specific genetic mutations that cause rapid, accelerated aging. This forest plot confirms the clock sensitivity. When analyzing the ZEMPS T24 deficient model, LivAge detects a massive positive age-dev beta coefficient. The LMNA mutant models behave identically, registering highly significant positive beta coefficients. These positive coefficients provide mathematical proof that the clock successfully detects accelerated biological decay, entirely independent of the animal's calendar age. Because LivAge accurately recognizes progeroid mice as biologically older, it is fully validated as a diagnostic tool for biological aging, setting the stage for its primary application, evaluating geroprotectors. The most critical application of the LivAge architecture is the rapid screening of pharmacological and dietary longevity interventions. If a geroprotective intervention is actually working, it must manifest within the model as a negative age-dev beta coefficient, indicating a reduction in biological age. To test this, researchers first examined the model's evaluation of dietary caloric restriction, a well-documented and historically validated intervention. This plot outlines dietary interventions. 8-month caloric restriction triggers a significant negative beta shift. Restricting methionine produces a similar transcriptomic age deceleration. Remarkably, these benefits are measurable even when offsetting the severe biological damage of a high-fat diet. The clock is equally adept at evaluating pharmacological interventions, particularly compounds like the mTOR inhibitor rapamycin. This final plot maps those pharmacological tests. Rapamycin yielded the most dramatic negative beta coefficient, providing mathematical proof of transcriptomic age reversal. Notably, this negative deviation occurred even while subjects were maintained on a toxic, high-fat diet. LivAge quantifies the exact magnitude of age deceleration induced by these geroprotectors, moving longevity science from theoretical assumption to measurable, actionable data. The architectural journey of LivAge relies on combining differential expression filters with Box-Cox and U-gene transformations to completely neutralize RNA sequencing batch noise. The result is a highly engineered, interpretably transparent model that tracks actual tissue function, contrasting sharply with the opaque mechanics of epigenetic clocks. The final logistical barrier to adopting this model has been the steep computational complexity of running these dual mathematical transformations from scratch. To eliminate this computational bottleneck, the researchers launched the LivAge web-based platform. This platform allows researchers globally to upload raw RNA sequencing matrices and instantly apply the trained ElasticNet model to their own data. Because the platform automatically executes the crucial variable transformations behind the scenes, it standardizes age-dev analysis across the entire biogerontology field. This accessibility democratizes rigorous, biologically interpretable age scoring, making it instantly available for any laboratory equipped to sequence RNA. By neutralizing transcriptolic noise, LivAge provides the architecture needed to evaluate and validate the next generation of geroprotective interventions.